Hey, AP Bio folks. Hope you guys are having a good week. I wanted to take some time here and go through some notes about Chapter 8, which is about metabolism. It's called Intro to Metabolism. So it goes through a bunch of things with enzymes and some ATP, some energy related stuff, all as a precursor to looking at cellular respiration and photosynthesis. So put together some skeletal notes for you guys, and I wanted to go through page 33, Thermodynamics and ATP, with you. So if you've got it downloaded, take a quick peek. Excuse me. The first bit you got there, there's basically three sort of things that we want you to get out of this. The AP board has agreed to, and these are the learning targets for this one. I want you to be able to explain how living systems adhere to the first and second laws of thermodynamics. So we got to understand the first and second laws. We have to look at how living systems apparently defy the second law of thermodynamics by increasing in order. And then lastly, we want to look at ATP and see how that works with exergonic and endergonic reactions. So a bunch of almost physics-ish terms here dealing with thermodynamics and ATP. So the first thing I'll show you is, this is from your book. I just pulled an image out of there, but it goes through and does a good job of looking at and explaining sort of what is happening in a really generalized reaction. So basically you start with something, you have a series of reactions till you get a product, right? And then essentially what we're having here is we're taking whatever it is and we are conserving that energy and we are just readjusting it with the help of an enzyme or two or three to get this thing to happen. And we get it to happen at a much quicker pace than is normally done. So the book gets into reactions, reactants, and products. So make sure you understand the basic language of that. And then we get into the first and second law of thermodynamics. Yikes, I'm going to struggle with that every time I say it. But the first law of thermodynamics, also known as the law of conservation of energy, basically states that energy cannot be created or destroyed in an isolated system. So essentially, we can't just randomly get it out of somewhere. It has to come from somewhere. And we can't get rid of it that way. It is transferred. It is essentially changed into another form, right? So in here, in the case the bear is eating it, it's going from chemical energy. He's going to create it within him to make ATP, and then he's going to use it to do work, all right? The second law of thermodynamics is that the entropy of any isolated system always increases, right? Entropy is basically a fancy way of saying disorder. So the thing about how organisms looks like they defy this, it basically says that sort of disorder or entropy decreases in an organism, but the universal's total entropy increases. So that's sort of the way that it looks. What do I mean when I say entropy or disorder decreases in an organism? Well, we become more ordered, we become more structured. We take and we make this ATP, so we actually organize this energy into a form, but on a whole, the universe's total entropy is going to decrease because of that. So it's sort of a little heady kind of thing to look at, but the idea of free energy is mathematically proven in this thing called Gibbs free energy. Now, this is a formula that you do not have to use. They took this out of the AP curriculum in terms of using the formula, but you still need to understand the concept of what Gibbs free energy says. And basically, it is a way of looking at and calculating out the amount of free energy in a system. So the formula is excuse me, listed at the top here, right here. Check, we got the formula. What do all those things mean? Well, the delta H, that is the change in enthalpy or total energy within a system. Delta S is the change in entropy or disorder, right? Depends on how you think about it. And then the basic concept is that in a spontaneous process, the delta G, or the free energy, is always going to be negative. So when you do the math, It will always equal a negative. If it's a negative number, it is a spontaneous process, right? That is the key. So a system must either give up enthalpy or have a positive T delta S, right? So the idea is, and the thing you need to get out of this is if the free energy is positive or zero, then the process is not spontaneous. If it is negative, then it is. And that is the whole gist of all of this. We're not going to go crazy, but Gibbs free energy is the idea. Realistically, it's almost better to call it available energy, more realistically, right? So Gibbs free energy is the available energy in a system, and it is calculated out. But the big thing to remember is if it is negative, it's spontaneous. If it's not spontaneous, it's going to come out as positive or zero, and that's the important part to remember. Now, when you look at how these reactions occur, this whole idea of endergonic, exergonic, what I would say to you guys is the term itself will help you to describe what is actually happening. So the word exergonic, if it's an exergonic reaction, that means, right, big E is energy. Energy is released, right? And it's spontaneous. That is a exergonic reaction. Energy is being released, okay? So if you look here, these would be your reactants, right? And what is happening then, this is up here, this is your free energy. In other words, those reactants, there's a lot of available energy. And what is going to happen then is that as the reaction, so this is the progress down here, Rxn is reaction. So as that reaction progresses, energy is going to be released into the environment right here. So there I got my energy. And then what I'm left with are some products, right? The amount of energy released, right? This is going to be, right, less than zero. So that is an exergonic reaction. Now, the other version is probably fairly clear, right? Instead of it releasing energy, it absorbs. So this would be an endergonic reaction. That means energy is required and it is not spontaneous. So again, free energy here. Here are your reactants. Energy is input into it, so you're adding energy to get your products. And the amount of energy here is going to be greater than zero. So these two types of reactions, when you talk about is energy required, is it spontaneous, is it not spontaneous, the key is to remember endergonic, energy has to be put in and it's not spontaneous, versus exergonic, energy is released and that's a spontaneous reaction. Okay, so that is the idea behind these things. Now, oh, my dogs. The whole key about this is a lot of this stuff that's being used or released is this thing called ATP, adenosine triphosphate. And we've heard about it before. I always call it the currency of life. It's the currency of energy. So anytime your body needs to do something, it has to spend this currency, which is ATP. So ATP is made up of 3 phosphate groups. That's the TP, the triphosphate. So we've got our phosphate groups. And then attached to that, you have the A of the ATP, which is adenine. And you have a sugar. In this case, it is ribose. So those 3 parts make up ATP, adenosine triphosphate. Now, once you start to make this stuff, there is a lot going on with what is there, how does it store energy. So there's this idea that the bonds between the phosphates and that ATP tail, so when you look at this portion right here, this tail, that is where a lot of this energy is stored, and it has to be broken up, right, by this idea. And we know the chemical reaction, right, is called hydrolysis. So we add water, we break this thing up right there between that second and third phosphate, And when we do that, the terminal phosphate releases a bunch of energy. So this release of energy comes from a chemical change to a state of lower free energy, not from the bonds themselves, but from changing the state of it. Often it's said that that bond contains the energy. It's kind of a generalized way to say it. But what we essentially have is ATP becoming ADP with that other phosphate, and then this energy is released by the breaking of that bond and changing it to a lower state of energy. So on this slide, it didn't quite come through on the one you guys have. It's missing parts B and C. But this is just an overview of this coupled reaction that we're talking about. So what it's trying to show you here is that if you had a single reaction where you took and converted to glutamate or glutamine, right here, these two are the reactants. They make this product of glutamine, and that gives you a free energy of positive 3.4 kilocalories per mole. So a positive number, right? Here is a reaction that is coupled with ATP hydrolysis. So we add, right? We take this, we add ATP towards it, we get this intermediate, then that intermediate, right? We add the same stuff, and now we get the reaction. So basically, if you do the math on this one, you have it's a coupled reaction. So we couple this ATP production or use with the combination of these, and we still get the same thing in the end. But now, because we've used that ATP as well, we have a positive, right, from up here, we have a positive 3.4 from these two to create this. But then we couple that with the ATP, which gives us this, and now we have a net free energy of negative 3.9, right? So which makes this a spontaneous reaction. So by coupling a reaction together, one that isn't and one that is endergonic, exergonic, it actually allows us to then create this coupled reaction to drive that so that overall it is an exergonic reaction, right? So coupling reactions works well, and there's enzymes that are going to make this happen. So, excuse me. The idea here is that we have this ability to have ATP drive transport, drive mechanical work, do all these things by taking that energy that is stored up in the ATP and using it to do things. So when we talked about proteins back in our biomolecule or macromolecule section of our chemistry notes, we were talking about how these transport proteins, if they are, depending on which way they're going, how they use ATP to actively transport things in one direction, right? So that ATP phosphorylation is going to cause that transportation of a protein, that breakdown. Down here, we have mechanical work. So this is actually inside of a muscle cell. There are motor proteins, actin and myosin, and basically they can do this movement, right? So this is actually a cytoskeletal tract. So the vesicle is going to have to move along, and it's done by sort of walking these little proteins along, right, to create movement and to get those vesicles where they need to go. So that ATP, again, is going to cause that reaction. The hydrolysis of ATP we're going to use that energy to create some kind of work, some kind of mechanical or transport work. So this whole idea, when you look at ATP, is it's this amazing thing, right, that when you have it, right, you use water to break it apart, that gives us the ability to do some kind of cellular work. We then can reload it, right? We take ADP, we re-add that phosphate, and we're going to learn about the process of doing this in our next bit. But that will then allow the reproduction of ATP. So this constant cycle of endergonic reactions, energy consuming, energy releasing, energy consuming, energy releasing is the key to life in terms of energy. So this ATP is pretty important to our life because it basically causes every reaction to happen. So the big question is, how do we make it? All right? And there are a couple ways. One way is called substrate level phosphorylation. Right? And this is sort of the simplest version. If you look at what we have here, we have the substrate, which has this extra phosphate group on it. We have ADP, and the substrate level phosphorylation basically says that we take the substrate, in this case it's called PEP. It's a long name, they simplify it to PEP, but it, using an enzyme, will give up that phosphate group to the ADP to create ATP in this byproduct of pyruvate. Now, your body can then go on and use this pyruvate in other ways, but this is substrate level phosphorylation, and this is a simple version. It doesn't make a lot of ATP. It doesn't have a way of making massive amounts for each one. It can just make that single ATP, but this can then be used to continue this process, right? So we're going to get into this whole idea of cellular respiration and how more is made. But substrate level phosphorylation is one way that it's made. The other way that it's made is this idea called chemiosmosis, all right? And then we have our mitochondria, our powerhouse of the cell, or a chloroplast. And basically what we're going to do is we want to trap hydrogen ions in the intermembrane space. So that folded membrane, we want to trap H's all through this area in that intermembrane space. And that will then create a concentration gradient that will drive the proton to flow back through a specific protein and create massive amounts of ATP. So when you look at it, that is the key. How can this lead to ATP production? Well, all these hydrogen ions have created a gradient. Now they're going to flow back through and produce massive amounts of ATP. That enzyme, if you guys remember, is called ATP synthase. So here is the gradient we've created. Here is the ATP synthase enzyme. These will now flow back through, and as they do, they will cause the phosphorylation, ADP, and the phosphate to join together to make ATP. So, and again, we're going to dig into this question, but there is a very specific way that this thing all happens. So, basically, the membrane is permeable to hydrogen ions. Hydrogen ions flow down, right, down the concentration gradient, much like water flows over a water wheel. As they flow through, they cause this thing to rotate, and as it rotates, that causes a change in the shape of ATP synthase, which is an enzyme, and that changing in shape powers the bonding of ADP and phosphate to make ATP. So that is the whole end product, is this is what we need to make massive amounts of ATP, right? Now, why? Well, there are enzymes involved in this process, and they're super important because they lower the activation energy of these reactions. If you talk to Mr. Dirksen, he'll probably argue they don't lower the activation energy, they give it a different pathway. It's basically saying the same thing here. But essentially, the way our book and the way the AP Bio sort of explains it is that without the reaction, there is an input of energy needed. That's the activation energy to make that reaction then go downhill to create those products. With an enzyme, right, the reactants are still the same, the product is still the same, but the amount of energy needed to trigger it to happen is lower, and we can then get the same products. So the free energy is unaffected by the enzymes, but the activation energy is going to be lower when there's an enzyme, which allows it to happen much cheaper in terms of energy, and it allows it to happen much faster. So enzymes are going to lower that activation energy by essentially making the products or the reactants find each other quicker, align themselves in the way they're supposed to, and happen at a much faster rate. So, and this is what I was just saying, in the enzymatic reaction, the substrate binds to the active site of that enzyme. That active site will lower the energy by making them line up properly. Imagine they had to be in a specific match, and if they didn't have an enzyme, they might be floating around and bumping into each other, and they may not find that match. So the enzyme grabs them, puts them together. So check, we can orient them correctly. It's straining the substrate bonds, so it makes them organize and either break or bond faster. It gives them a more favorable microenvironment for the bonds to happen or to break. And it covalently bonding to the substrate to hold everything tight. So it'll make it happen so much faster. Without it, we would be a very different organism because we would need these reactions to happen at a much quicker pace than they do. Now, the last few slides here are just from your text. These are pictures that explain or show a series of them, how these reactions are occurring with enzymes. Remember, this enzyme is very specific. The active site is very specific to a specific set of substrates. If they are the wrong ones, it won't work. If the enzyme gets misshapen because of overheated or acid or base kind of causes some misshape, this thing is not going to work. So those substrates will bind with weak interactions inside of this, so it's all set up properly. Once they are there, then that is where that activation energy is going to be lowered. It speeds up the reaction. So here you can see now, instead of being vertically set, now they are created this way. So it converted them to whatever products you want to get. Once you converted them, they are released and that enzyme is freed up. It was not changed. Any kind of conformational changes were not permanent. So it now can go and do work again until it's worn out. So your body, rather than have these two bump into each other and figure that out on their own, when I say figure that out, I mean have them bump into each other on their own and work, this enzyme puts them together. I always used to think of it as like speed dating for substrates. Essentially, these two were meant for each other, but they maybe would never find each other without that speed dating to make it happen. A little silly, but makes sense. So the thermodynamics and the ATP, the stuff with ATP can get kind of heady. I didn't get into what the specifics of ATP production are because that's going to come next. I want you to focus for this stuff on sort of that idea of free energy. So really make sure you go through the textbook on this section. It can be a little funky. It's a lot of physics, to be honest. But the basic premise is you need to understand those exergonic versus endergonic reactions and to see which ones are going to produce energy or use energy, and then how enzymes can make that a little less by decreasing activation energies or making them find each other a little quicker. So that is kind of our general overview of the Chapter 8 stuff. Again, spend some time with that reading guide, and that will lead us into next week where we can look at the specifics of energy production with cellular respiration and then photosynthesis the following week. All right, you guys have a good one. We'll see you in class.